So thanks for having me. Today, I'm going to be speaking about some of the work that I did for my master's, which was looking at a really incredible but threatened fish called Pugnose Shiner. Um, and I did just want to give a shout out to AFS and especially the Ontario chapter, because back in 2018, this was the first conference that I ever attended and ever presented at. And I was super nervous, maybe you can tell from the photo, but it's really exciting for me to be back here, at least in a virtual setting, to present some real results this time and to connect with some people. So a huge thank you to Craig and Sarah for some incredible work organizing this. It's been great to be back. So without further ado, let's move on to the star of the show, which is of course Pugnose Shiner, otherwise known as Notropus anogenus. And for those of you that haven't had the privilege of meeting this little guy, they prefer clear, cool, highly oxygenated, highly vegetated sites. So sites with really excellent water quality. And their distinguishing feature is this highly upturned mouth, which allegedly makes them look like a pug. To me, it kind of just makes them look a little bit grumpy. And these days they have a lot of reasons to be grumpy because they are in fact a threatened species under the Species at Risk Act. So this map shows the distribution of Pugnose Shiner in Canada. So in red, you'll see those dots there. And what's really clear right off the bat is that they're found in these really kind of small, isolated populations, just in Ontario and specifically Southern Ontario, including a really robust population in the St. Lawrence River that I wanted to point out too, just because we had a lot of talks yesterday about the St. Lawrence and the Thousand Islands region, which is where I did a little bit of other work for my master's. So as we all know, and as Steve Cook talked a little bit about, freshwater fish are having to contend with multiple stressors in their environment. For Pugnose Shiner specifically, one of the major threats seems to be turbidity. But it's not just the individual impacts of these individual stressors, but of course, how they interact with each other to potentially exacerbate some of these impacts on these already vulnerable species. So for my research, I decided to hone in on an emerging threat, climate change. And I asked a whole bunch of questions related to their physiology, their growth, their metabolism, and their distribution as well. But today I'm going to be focusing specifically on how elevated water temperatures affect the physiology of Pugnose Shiner. So to do this work, we did rely on a whole bunch of lab experiments. And as you might imagine, when you're working with a species at risk, getting enough individuals into the lab to actually run experiments on them can be challenging because you don't want to have a negative impact as a researcher working with these already threatened species. So we were really, really lucky in that we had access to this pond rearing facility at the State University of New York, Cobleskill. So back in 2015, one of the students there actually it was a student in collaboration with other people. It wasn't just a rogue student. They decided to stock this pond with 65 adult Pugnose Shiner. And then over the next couple of years, the population in this pond actually proliferated to the point where we arrived in October 2017. And we were able to sample like hundreds of Pugnose Shiner in this pond and get out of there pretty quickly. So we're super grateful to the folks at SUNY Cobleskill for allowing us to use these fish and for having kind of that vision for this pond rearing facility in the first place. So once we got them back across the border and into Canada, we separated them into juveniles and adults based off of size. And I focused on the juveniles. And we were specifically interested in looking at how juveniles could respond to elevated water temperatures over these longer term developmental periods. So we held them under 5 different temperatures for 4 months before running a whole bunch of various metabolic trials. So one of those tests, actually, never mind. We looked at those 5 different temperatures based off of their summer thermal range in Ontario. So this graph is just showing you the water temperature at sites where Pugnose Shiner were captured by the DFO between 2002 and 2016. So what you can see is that kind of that lower limit and the upper limit that we selected really kind of captures that summer thermal distribution of Pugnose Shiner. Obviously with climate change, that would push it a little bit higher. So the first metric we looked at, and one that's widely used in the literature today, is a metric known as the critical thermal maximum, or CT max. And I included a fancy definition up there, but mechanistically what that looks like in a lab is putting a fish in a tank, slowly increasing the temperature, and then at a certain point they lose equilibrium. And so we often use this as a measurement of their thermal tolerance. So this is a picture of what our system looks like. We actually ended up running 2 fish at the same time. And to get at some of these potential interactive effects, we decided to test CT max under both high oxygen or normoxic conditions and also low oxygen hypoxic conditions. So here are some of our results. So on the x-axis, you'll see those 5 different rearing temperatures. And then on the y-axis, you'll see the CT max, or that upper thermal limit. And what's really clear right away is that across every single rearing temperature, CT max increased, which is what we would expect. And it's pretty common in the CT max literature for fish to show some sort of acclimation capacity to different temperature treatments. But what was really cool for us is that if you look at the slope of this relationship, you get a value of 0.61, which means that for every degree increase in their rearing temperature, we saw a 0.61 degree increase in their CT max. And that's actually relatively high. In one meta-analysis from 2000 across 21 species, they found an average CT max of 0.38. So it seems as if Pugnose Shiner, this species at risk, might have a relatively high acclimation capacity to warming waters. The key point being that this is under normoxic conditions. Oops, for some reason I can't click through. Oh, there you go. So what happens when we measure CT max under acute exposure to hypoxia? Well, then it looks a little bit different. So in orange now you're seeing the CT max measured under low oxygen conditions. And what you can see right away is that across every single rearing temperature, the CT max under low oxygen is lower. Furthermore, if you look at those kind of 3 intermediate temperatures, we saw no increase in their CT max, despite the 6-degree increase in their rearing temperature. So something is really happening under hypoxia that's limiting this thermal tolerance. Obviously, something else is also happening at 28 degrees. We think it could be related to metabolic depression or greater reliance on anaerobic metabolism, but that's something we weren't able to capture within the study. So in addition to looking at CT max, we decided to look at another behavioral threshold that occurs before fish reach that upper thermal tolerance. So this is called agitation temperature, or TAG. And it was first described by my supervisor and another colleague in 2015. And essentially they noticed that as you kind of increase the temperature during a CT max trial, the fish just isn't going to sit there waiting for things to happen. It's going to start to dart around and look for an escape. So we can measure this and we can quantify this as the onset of thermal stress. So here are results for agitation temperature. And again, what we're seeing is that in the hypoxia trials, the agitation temperature is lower. I did want to also point out that it's very likely that we overestimated the agitation temperature in those hypoxic trials. So if you look at 25 degrees, for example, you'll see that they are already exhibiting agitation temperature at that home value, which means that oxygen itself was causing a pretty significant stress. And so it's likely that those temperatures are overestimated in the hypoxic trials. So overall, what we found is that Pugnose Shiner, perhaps hopefully a little bit, have a relatively high acclimation capacity to elevated water temperatures. However, acute exposure to hypoxia limits their ability to tolerate those high waters and also affects some of these behavioral thresholds as well. So it kind of highlights the importance of looking not only at individual stressors by themselves, but how they might also interact in the wild to give a more ecologically meaningful picture of what's happening for some of these imperiled species. And of course, one of the focuses of my research, because we were working with a species at risk, was to try and figure out how we can use this information for management strategies. So recognizing the significance of these interactions between stressors and recognizing also that hypoxia is potentially easier to mitigate by, for example, preventing nutrient influx into systems. By mitigating hypoxia, we might be able to buffer some of the negative impacts of climate change. And potentially as well, if we wanted to look at a more kind of adaptive management response, if we're focusing on reintroduction efforts, we could either look at populations that already display higher thermal tolerance, or we might implement captive breeding programs under different temperature regimes to increase the success of those introduction efforts in the wild. So with that, I wanted to say thank you to all of my collaborators, supervisors, and of course to Pugnose Shiner as well, who I've had the immense privilege of getting to learn from over the last couple of years. Thank you.